Alzheimer's disease, or AD, is the most prevalent form of dementia, contributing to 60 to 70% of cases worldwide. According to the World Health Organization, over 33 million people are affected globally. Currently, AD is diagnosed when individuals experience significant memory and cognitive challenges that disrupt daily life. With no cure available, early diagnosis is crucial to managing the disease's progression. Researchers are now exploring innovative methods to detect AD earlier, potentially reducing the social, emotional, and financial impacts on patients and their families. A groundbreaking study has unveiled a promising approach to diagnosing Alzheimer's disease earlier by analyzing biomarkers in blood. This research, led by Dr. Brandon Mahan from the University of Melbourne, aims to identify AD before cognitive decline becomes apparent. The study, published in Metabiomics, highlights the potential of a blood test that could revolutionize AD diagnosis. By detecting the disease earlier, patients could benefit from timely lifestyle changes and medications that may slow disease progression. This advancement could also expand eligibility for clinical trials, offering further medical benefits and advancing research in the field. The research team, comprising analytical geochemists and neuroscientists, has adapted techniques from cosmochemistry to search for early biomarkers of AD in human blood serum. Originally developed to study the formation and evolution of celestial bodies, these highly sensitive techniques have been repurposed for medical research. By applying inorganic analytical geochemistry, the team has opened a new avenue for diagnosing Alzheimer's disease. This interdisciplinary collaboration between the Faculty of Science and the Faculty of Medicine, Dentistry, and Health Sciences at the University of Melbourne exemplifies the innovative approaches being taken to tackle this global health challenge. The researchers focused on assessing the relative levels of potassium isotopes in blood serum. In a pilot study involving 20 samples, 10 from healthy individuals and 10 from AD patients, the test demonstrated potential for early diagnosis. Our minimally invasive test assesses the relative levels of potassium isotopes in human blood serum and shows potential to diagnose AD before cognitive decline or other disease symptoms become apparent, said Dr. Mahan. Unlike protein-based diagnostics, which can degrade during storage, this test evaluates an inorganic biomarker, ensuring sample stability and scalability for widespread use. Early diagnosis of Alzheimer's disease could have profound implications. It would allow patients and their families to take proactive measures to manage the disease's progression. Earlier diagnosis would enable earlier lifestyle changes and medication that can help slow disease progression, Dr. Mahan explained. This could also alleviate the emotional and financial burdens on families, providing them with more time to prepare and adapt. Furthermore, early diagnosis could increase patient eligibility for clinical trials, potentially leading to new treatments and therapies that could benefit future generations. While the initial results are promising, significant work remains to determine the ultimate utility of this novel diagnostic technique. Co-author Professor Ashley Bush from the Florey emphasized the need for further research. Our blood test successfully identified AD and shows diagnostic power that could rival leading blood tests currently used in clinical diagnosis, Professor Bush stated. As the global population ages, the incidence of Alzheimer's disease is expected to rise, doubling every 20 years. Continued research and development are essential to address this growing challenge and to refine the potassium isotope test for clinical application.